I'm Dr. Aisha Ambreen in continuity with the cell signaling chapter. Today, in lecture number 6, we are going to discuss about the signaling pathway of JAK-STAT signaling, tyrosine kinase signaling, and SMAD pathway. TGF alpha or TGF beta, which follow the SMAD pathway. JAK-STAT signaling pathway. JAK is abbreviation of Janus kinase. The Janus kinase signal transducers and activators of transcription, STAT protein. Intracellular signaling pathway is activated in response to a large number of cytokines, growth hormones, and other hormones. It is involved in numerous functions within the body. This signaling pathway is stimulated by the interaction of ligands with their receptors, then causing the receptor activation. In this pathway, only the receptor, not only the receptor binding takes place, but the receptor itself also gets activated. Then the transphosphorylation of receptor associated JAK molecules takes place. After that phosphorylation of the receptors, after that it leads to the phosphorylation of tyrosine residues on the receptors, and then phosphorylation of downstream signaling molecules called STATs. These STAT molecules are cytoplasmic proteins which pass to nucleus and allow the transcription of specific genes. Diagrammatic representation here. Any cytokine or hormone which is going to bind on the receptors, it is going to activate the receptors, and both of the dimer forms of the receptor, they will transphosphorylate each other. This side of JAK will transphosphorylate the other receptor's JAK, and this JAK will phosphorylate the first JAK molecule attached to the receptor one arm. Second thing that occurs is after phosphorylation, these JAK molecules, they also phosphorylate the receptors specific residues, and after their phosphorylation, STAT protein coming from different cytosolic source, that STAT protein binds to the phosphorylated receptor part and gets phosphorylated by the help of JAK molecule. So JAK molecule, after transphosphorylation, not only phosphorylates the receptors, but it also phosphorylates the STAT molecules at second position. After full phosphorylation, STAT molecules, they are separated. These STATs dissociate from the receptor and then dimerize with each other. After dimerization, these STAT molecules, they become a transcriptional factor and translocate to the nucleus. This is another cytosolic representation of JAK-STAT signaling pathway, in which cytokine is attached. Cytokine as a ligand is attaching, and the same cascade is taking place. After that, the STAT is dimerized, and the dimerized STAT will be transported or translocated towards the nucleus. On the nucleus, the STAT dimerized form is going to enhance the gene transcription. Receptor tyrosine kinase. Receptor tyrosine kinase is another kind of receptor family. These receptor tyrosine kinases are another class of receptors revealed to show diversity in their actions and mechanism of actions. For example, the general method of activation follows a ligand binding to the receptor tyrosine kinase, which allows their kinase domains to dimerize. Then next, this dimerization invites the phosphorylation of their tyrosine kinase domains, which allows intracellular proteins to bind the phosphorylated sites and become active. An important function of receptor tyrosine kinase is their roles in mediating growth pathways. Keep in mind these growth pathways not only in the normal growth but also in proliferation. The tyrosine kinases also play important role, as well as they play also a role in the cell mediated program death, or you can say the cell death is also programmed after the tyrosine kinase activation. So different pathways, there are variety of pathways that are following the tyrosine kinase and in their actions. One of the pathways shown here, the tyrosine kinase, as you can see, it has 4 arms. 2 are extending out of the cell membrane surface and 2 are spanning within the cell membrane and inside the cytosol. Mainly the tyrosine residue in the tyrosine kinase receptors is phosphorylated. That's why its name is given tyrosine kinase. This tyrosine kinase is also acting as a enzyme after the binding of any ligand, and it activates other enzymes. For example, here it is activating IP3 kinase. IP3 kinase is the enzyme which is going to phosphorylate the IP3. IP3 is inositol triphosphate, which is the product of PIP2 and originating from the membrane. It converts into IP4. Similarly, IP6 can be converted into IP7, and they have further many other functions, AKT and PH, which are going to again bind with the phosphatidyl inositol derivatives, and their further cascades are depending upon the requirement of the cell. Either they are going to increase the cell growth, or they may lead to the apoptosis. This is another diagram for tyrosine kinase, in which the receptor is bind to the other cytosolic proteins, like here is IRS. This IRS has 2 SH domains here. The SH2 domains with which can be phosphorylated, and further they phosphorylate further protein or effector proteins. Similarly, here another tyrosine kinase is shown, which can bind to the SHC or GRUB2 and RAS proteins. These are all the cytosolic proteins which are mediating different cascades and pathways. One of the cascade of activation of this RAS protein is showing here. For example, the growth factors like tumor growth factor alpha, TGF means tumor growth factor alpha, EGF epidermal growth factor, and just like these growth factors, there are many other factors which are going to bind to the tyrosine kinase receptor. And tyrosine kinase receptor, after activation, it attaches RAS molecule or RAS protein. It activates RAS protein after phosphorylation, and RAS protein activates RAF protein, and which activates MEK, and MEK is going to again phosphorylate or activate ERK. So all of them, they form a cascade. This cascade of CRAF, MEK, ERK dependent effects show proliferation, differentiation of cell, survival of the cell, adhesion of the cells to the other bodies, or interadhesion, angiogenesis in the case of cancer, or some proliferation or mobility of the cell. So anything related to the growth and differentiation is carried out by this pathway, and many other more pathways also. In case of apoptosis, CRAF, I am not going to deliver whole details here, because I'm just trying to give you a main idea that how the phosphorylation of one protein that accounts for the phosphorylation of many other rest of factor proteins, and all of them, they turn into sequence, either leading towards differentiation or proliferation, like here in case of apoptosis inhibition, the CRAF that inhibits the BAD gene expression, and it increases the BCL2 cytochrome C release mitochondrial expression to increase the growth. Similarly, here GPCR, which I have already explained about them, G-coupled protein receptors, they bind to chemokines, interleukins, hormones. All of them, they will become familiar to you when you will study in detail in different chapters. So interleukins or chemokines, they are mostly produced in response to some immune response, and they are going to activate the G protein, and G protein is going to activate phospholipase C, then it is going to activate protein kinase C. So protein kinase C, you have studied it. Among all other functions, protein kinase C also activates the CRAF protein. CRAF has many other functions like this one. Likewise, adenylyl cyclase activating protein kinase A. In the initial lectures, protein kinase A activation and its amplification was explained. So one of the amplification of them is shown here. Here I have summarized all of these functions. For example, you focus on the second messengers, then you can easily understand the pathways. For example, in cyclic AMP, glucagon is the ligand and the protein substrates. And in the case of cyclic GMP, protein kinase G here, and in the case of colmodulin, in case of G protein coupled receptors, cyclic GMP is the second case. Then calcium colmodulin, which was discussed, and then diacylglycerol, which can also activate protein kinase C, and IP3 activating with the help of calcium colmodulin. And the last, insulin, that is going to act with the help of tyrosine kinase, and protein serine threonine kinase is also activated by the help of proteins activated by tyrosine kinase. Now comes the SMAD pathway. Here the TGF beta SMAD pathway is shown here. In this pathway, the first step in this pathway is the binding of the ligand to the specific TGF beta type 2 receptor. The binding recruits TGF beta type 1 receptor also. Being a serine threonine kinase, type 2 receptor phosphorylate TGF beta type 1, thereby forming a dimer. Type 2 and type 1, they become dimer. Type 1 receptor in turn phosphorylates SMAD protein. The phosphorylated SMAD protein is then translocated to the nucleus where it regulates the gene expression. Here the TGF beta in this pink color is showing. It acts as a ligand and it attaches first with the type 2 receptor. Type 2 receptor after phosphorylation, or it also dimerizes with the type 1 receptor. Type 1 and type 2 receptors after becoming dimerization process, then they phosphorylate, and their phosphorylation also induces a SMAD protein to get phosphorylated. After phosphorylation, SMAD protein binds with another SMAD to transport towards the nucleus where it enhances the transcription. This was about the SMAD proteins.